Dear audience, welcome to today's webinar on conversion prediction of an enzymatic esterification. My name is Andreas Liese and I am the head of the Institute of Technical Biocatalysis at the Hamburg University of Technology in Germany. The lecture today will be given by Jakob Müller, who studied bioprocess engineering at our technical university and is working as a PhD student in my institute since 2008. In our institute, chemists and biologists and engineers work actively together. We do offer to companies research and development in the fields of kinetic and thermodynamic analysis, mathematical simulation of biotransformations and bioprocesses thereof, optimization of biotransformations and process development up to multi-gram to kilogram quantities. If you're wondering what enzymatic biotransformations are, one short example. The compound to the top right, adenosine triphosphate, is the energy currency in each of your bodies. You produce half of your body weight per day of this compound. This means if you're weighing 70 kilogram, you're producing 35 kilogram per day. The only luck that you have, that you're not depressed by this weight, is that the steady state concentration is just around 150 grams, because this compound is synthesized and consumed all over in reaction sequences. This example demonstrates that nature's chemical technology enables the production of multi-kilogram of chemical compounds. Each one of you is able to this. So why not use this technology for industrial production of chemicals?
Thank you, Professor Liese, for this kind introduction. Hello and welcome also from my side. My name is Jakob Müller and I will give you today an overview about the conversion prediction of an enzymatic esterification with a Reactor 45M and IC Quant. This work is a cooperation between the Institute of Technical Biocatalysis at Hamburg University of Technology with the two companies Evonik Industries and METTLER TOLEDO. Here you can see the outline of my talk. First I will give a short introduction into the reaction system, the fatty acid esters. Then I will show you our reactor type bubble column to produce the fatty acid esters. Then I will come to the results section. We measured kinetics in this system with no additional offline data, only by acquiring the FTIR data. And then we also made a conversion prediction with the chemometric module IC Quant in a bubble column reactor. At the end of my talk I will give a short summary. What are fatty acid esters? These are esters from fatty acids and alcohols. You can combine an acid or fatty acid with an alcohol via a biocatalyst to an ester, and also water is formed during this reaction. There are 300 till 400 fatty acids in nature, and there's also an unlimited amount of alcohols available, so we have the possibility with this reaction to form a wide variety of products. Important thing here, fatty acids are often unpolar, and this is an easy possibility to combine these unpolar molecules with polar alcohols to form bipolar molecules known as surfactants or emulgators. Another important thing here is that fatty acids normally derive from nature, so it's possible to get these molecules by renewable resources, and this gives also the possibility to form sustainable products at the end. For which purposes fatty acid esters are used? One example is biodiesel. So biodiesel is in fact consist of fatty acid esters. So I use as energy source here. Another example is the cosmetic field. In the cosmetic products, these esters are used as emulgators or directly as oil components, and emulgators are used to combine the oil and water phase in these systems to make it more stable. And a third example here is food. So also in food applications, emulgators are used to make the product more stable. We want here concentrate on the fatty acid esters for cosmetics. So here are two examples. On the upper part you see a combination or an ester combination of sugar with a long chain fatty acid, and the sugar molecule here is the polar molecule, the fatty acid molecule is the unpolar molecule, so you have here bipolar molecule, emulgator, which is used in creams and so on. On the other hand, you have a very unpolar molecule which consists of two unpolar molecules, so this is more a molecule which is used as fat or oil component in these systems. How are these products produced? So there are chemical ways to produce them, and there are also enzymatic ways. On the left side you see a conventional system. The raw material is fed in the reactor. The reaction takes place at over 200 degrees Celsius. Problem here is you get a lot of side reaction, then you need a complex downstream processing, desaturation, bleaching, drying and filtration until you can have a good quality and can pack your product. The problem here is you see the color, so a lot of energy is needed and also a lot of waste is produced. So there are ways with enzymes, with biocatalysts, to produce this at a lower temperature. You don't need that much energy, and you don't have any side products, so you don't need any downstream processing except the packing. So you also don't have any waste, and this makes this process competitive to the conventional processes. So we spoke about biocatalysts. So what kind of biocatalyst we are using? We're using the Candida Antarctica lipase B. It's a widely used enzyme. It's adsorptive bound on a porous metacrylic resin, and it has a wide substrate spectrum, and compared to other lipases it has no boundary layer activation. This is the reactor type we are working with, the bubble column reactor. This is only a column where we feed in from the bottom a gas stream, for example air or nitrogen, and this reactor type is only low energy input because we don't have any stirrer inside, and the gas has two tasks. So on one hand you need to mix the substrate in the system, and this is done by the gas stream, and another task is the water removal. And why this is important, due to the reaction, because water is formed, and by removing this water in situ you are shifting the equilibrium of the reaction to the product side, and so you will end up with a higher product concentration. But this reactor type is also good for the production of high viscous products, so we have here access to new substrates, high viscous substrates, and so we can also produce high viscous products. The reactor concept is shown here in the pilot scale with a total amount of 120 liter, and the figure gives an impression about the reaction velocity, so how fast a specific conversion is reached for different reactor types. The red line, the stir tank reactor, the purple line the fixed bed reactor, and the rest of the lines different bubble column reactors. And it's obvious that the bubble column reactor performs this reaction faster. So there's a factor of 2.8 between the bubble column and the conventional used fixed bed reactor, and this leads to an improved space-time yield for the production process. And in this reactor type in a lab scale, we want to integrate now the Reactor 45M. This Reactor IR is equipped with an RTR probe, so an alternative to transmission probes, and also has a new fiber probe technology for efficient light guide, and the goal is to measure online direct in the reactor. Some words to the RTR technology. You can see in the bottom left picture the tip of the probe, and in this tip there's a diamond crystal, and you see the right picture the IR energy comes from the light source, goes to the crystal, and in this crystal the energy is reflected, and during the reflection the light is absorbed from the molecules which are directly on the crystal, and then the light goes back to the detector and is recorded there. Here you can see the spectra you get, so the absorbance over the wave number, and this is the typical MIR region, and this is over a reaction. You see a lot of changes over the time at the end, and you can plot this perfect in a 3D manner, and there are a lot of changes, and you can imagine that this is perfect for analysis. So this is a scheme how it looks at the end. So we have our reactor type bubble column, and in this reactor type we have our FTIR ATR probe, and I will repeat, we have additionally our biocatalyst inside, so a solid. We have our gas bubbles, so a gas phase, and we have a substrate mixture up to 2 phases. At the end we can work in a 4-phase system. Now I will come to the results part, the kinetic measurements with Reactor IR 45M without any offline data. What is the motivation? We want to measure the kinetics without any reference analytics, only by FTIR, by plugging in the FTIR in the system, measure, and then we get our data about the activity of our biocatalyst. Which system we are using? We have here myristate, this cosmetic oil with a low viscosity at 10 millipascal seconds. It's a one-phase substrate mixture, so the substrates are miscible in each other. Here you can see the spectra. The red spectra is only the acid, so you see a big peak where normally the acid absorbs, and the blue curve is the ester peak. So you see that they have 2 peaks which are not on the same level, so you can follow the reaction only by looking at the changes of these 2 peaks. And how does it look like? What does the program gives us? This is what you can see here. Here's the spectra over time. You see a lot of changes, so the acid is decreasing over the time, and at the same time at the left you see the ester is increasing, and now you can look at the, for example, at the peak area or the peak height. You can choose between these things and look how these parameters are changing over the time, and program gives you so-called trends, and these are the trends what the program gives you by looking at the changes over the time, and you can define your trends here on your own. And here you see now the change of the peak height on the red line of the acid, on the blue line the peak height of the ester, and the one is decreasing, the one is increasing. And now you can, by looking at the slope of this, you can calculate an activity. So here you can see the results. We calculated the specific activity in units per gram for different experiments. We will write here the molar ratio, so the starting amounts of the acid and the alcohol, and the activity was calculated based on the spectra, so the peak height of the acid changed over the time. And here we observed a maximum at 0.4 gram acid per gram alcohol, but now we have to prove if this is in the right range. And when we compare this with the acid values, or the activities determined via acid value determination, we see that we are very close. So the activity determination is quite good also with the FTIR measurement, and we need only half of the time for the collecting of the data. So the initial rate measurements worked out quite well, where we had only small changes and we had no influence of peaks on each other. But you can imagine if we would make a whole reaction, that there are possibilities of peaks which are near each other, that they have an influence on each other. For this problem there are solutions. On the left side, the gray curve is a mixture of the acid and the product. You see that the peaks are overlapping, and there are possibilities by pre-processing of these curves by the second derivative to separate them, and then you're able to have a look also on whole conversion curves, not only at the initial rate measurements. And this is what we observed for a synthesis of myristoyl myristate in N-heptane, and this is the conversion over time, and the blue dots is the acid value, the offline data measured by titration. And the conversion is at around, end conversion is at around 90%, and the FTIR conversion was calculated by change of the peak height over time after pre-processed, after second derivative of the spectra. And you see we have a very good correlation, so with the right pre-processing we are also able to follow the right conversion without any offline calibration. So what are the outcomes of this chapter? It's possible to make activity tests, so initial rating, without offline analytics. But for sure we have to say to this, you have to adapt this to your system, so it works not for every system. You need specific peaks which are changing and so on, but in principle it works. And you can also record whole conversion curves, but here also you need the appropriate data pre-processing. Now I come to the next topic, the conversion prediction in a multi-phase system. Now we don't want to look only at specific peaks. Now we want to take the whole spectra into account, make a chemometric model, and then use this for new experiments for an external validation. So again, here you see how this spectra look like. On the left side you see a conversion over time curve, and at the same time you see the spectra over time, and you see that over the conversion a lot of things are happening, so you have perfect situations for appropriate data processing and data analysis. How does this look like, this multivariate data analysis? First of all, we need to create a model. For this we make an experiment and take offline concentrations so that we know concentrations at every time. Ideally we can fit also the experiment. At the same time we're taking spectra from the system, so the whole wave number range. And this two data we feed in a chemometric model via multilinear regression and PLS solver. And this model we can use then for prediction of new experiments. This is called external validation, and for this we will take a new experiment with new spectra, feed this spectra into our model, and then the IC quant here give us our concentrations. Only a few words to the multiple linear regression. It's quite similar to the linear regression, only that you have not only a vector, you have a data matrix from the IR spectra, and you combine these with your measured offline data by a regression parameter and the deviation of the measured data to the regression, and you solve this by the method of partial least squares, and you try to minimize EY. And the good thing is you don't need to do it by your own. This is done by the software and the IC quant module for you. So, and this is a screenshot of the IC quant module. On the lower right part you see the spectra viewer, where you can see how the spectra look like over the whole wave number range, and there you can define a region which is taken into account for the model, and then you can combine spectra with conversion or with concentration information you took offline. This you can do in the upper left part, and with this two information, these are enough for create a model. And then it gives you an actual versus predicted plot, so that you can see if the model is accurate. So it's used on their own spectra which were before used for this model. And you can see here this model is quite well. You only have 16 points and 60 measurements, and it can calculate all the spectra quite well. And you can see on the right there are also possibilities for data treatments like baseline correction, smoothing, the normal things you can use for data analysis, and these are possible to also to use for this model creation. And this is the result here. You see our experiment for the calibration. We have a conversion time plot here, and the white dots are the measured data by titration, which were fed in with the FTIR data and formed to a model, and then the same experiment was predicted. So on this worked quite well, which was not surprising. But then we did two additional experiments, you see on the right side, and also the symbols are the offline data, and the curves or the small dots are the predicted data, and there you can see we are very exact. So also the external validation works, so for new experiments we can predict the reaction. Then we did a special experiment. We did 3 experiments with different starting conditions. We will write the molar ratio of acid to alcohol from 40 to 60, 50 to 50 and 60 to 40%. And all these experiments we monitored via titration and at the same time via FTIR. And all these 3 experiments we fed to a model and created a model. And yeah, it was surprising that we could predict all 3 experiments later with the same model. So 1 model for 3 experiments. This is interesting because we have within this model like overlapping information. So you have the acid value 80 has 3 values with 3 different data, but this model can calculate this and handle this. But we were happy then. We made a new experiment, an external experiment, which was not fed into the model, with additional starting conditions in between. It was around 55 to 45 molar ratio. And also this experiment, this model was able to predict. And I think this is a good example how flexible the data analysis is, how robust it is, or it can be when you do it appropriate. And here we calibrated the system to a wider range of starting conditions, and this is able to be used flexible for different experiments, not only for these experiments which were it based on. With this result I will close my talk. I will give you a short summary. First of all, I showed you our reaction system, the enzymatic production of fatty acid esters for cosmetic, and I also showed you a new reactor type with improved space-time yield for the production in this field. Then we combined the FTIR technology with this reactor type and reaction system, and we measured the kinetics without the offline concentration data for fast and easy results. The FTIR was used in this multi-phase system reactor with solid catalyst, gas phase and a liquid phase or 3-phase system, and we used successfully the IC quant prediction for this reaction system in this reactor type. So now I'm at the end of my presentation. Thank you for your interest, thank you for hearing, and it was a pleasure for me to be here, and I'm very happy to answer your questions.